Thank you for joining this session about biology. This webinar will be covering the first two sections of Chapter 11 in the biology textbook. This is about the introduction to genetics. It's pretty dense material, so I split it in half. So next week we'll be covering the other half of the chapter. If you're here live, please participate in all of the quick poll questions that you'll see on the screen. No one can see your individual answers, just the group's results. So if you're not sure if your answer is right, that's okay. Nobody's going to see what you said. And all the questions on here are just for practice. So they're not submitted anywhere. They're not for grading. If you're watching the video later, you don't have to do them. Just follow along. And if you are here live, you can definitely ask questions. If you're on a computer, you'll have a speech bubble icon. If you're on a phone or a tablet, you'll have a question mark icon. You click on that and you'll be able to type questions to me. And definitely ask questions about anything that you need me to go back and repeat, anything that I didn't explain well enough, anything that you have other questions about, or anything from this lesson that you have questions about that we could go over at the end. This is your time to ask questions of the instructor, so definitely don't hesitate to do that. Okay, so Chapter 11 again is Introduction to Genetics. So all of an individual organism's DNA came from its parents, but each individual has a different combination of DNA from the two parents, which gives them different traits, such as these different colored kittens that came from the litter. And also this litter of different colored puppies, because they are too cute to leave out. The study of the way that traits are passed down from one generation to the next is called genetics. When studying inheritance, we study the work of Gregor Mendel. He had sets of pea plants with a variety of traits, and bred plants with some traits, two plants with different traits, and then documented the traits inherited by the offspring. And this taught us a lot about how different genes are inherited. One conclusion that Mendel came to is that information passed down from parents to offspring is passed down in genes which control the traits that the individuals have. For example, all of his plants had genes that controlled the color of the seeds, and they inherited those genes from their parents. And there are different versions of the genes. Different versions are called alleles. For example, some of the seeds in Mendel's plants were yellow, some of the seeds were green, and some were gray. So he was working with three different alleles of the gene that controlled seed color. Another important conclusion from Mendel's work is that often when there are two different alleles present in an organism, one of them is expressed as a trait that you can see, and the other is hidden. The allele that is expressed is called the dominant allele, and the one that is hidden is the recessive allele. For example, in Mendel's plants, if a plant received the allele for green seeds from each of its parents, the seeds were green. But if the plants received a yellow seed allele from one parent and a green seed allele from the other parent, the seed of the offspring was always yellow, never green. The allele for the yellow is dominant and green is recessive. So if they're in the same plant, you'll always see yellow and you'll never see green. We're going to be talking a lot about dominant and recessive alleles and doing diagrams with them. And when we do this, the dominant alleles are always given capital letters and recessive alleles are given lowercase letters. And we will be using the following examples for color. So when we're talking about the color of the seeds, we're going to use a capital A for dominant and a lowercase a for recessive. And with this specific version, the colors, yellow is dominant, so that'll be a capital A, and green is recessive, so green will be a lowercase a. That may seem kind of strange. Like, why wouldn't we just put Y and G for yellow and green? But because it's the same gene, we need to use the same letter. So we use the same letter and use a capital for dominant and lowercase of that letter for recessive. And it doesn't really matter what letter we go with, so I picked A. An important concept for understanding inheritance is the segregation of alleles. Every individual has two of every gene, one from each parent. But then when that individual reproduces, it goes to make reproductive cells, those two copies of the gene split up so that the egg or sperm only has one copy. So most cells in the body of the plant or the animal or the person have two copies of the genes, but when it makes eggs or sperm, they're split up, so each of those cells only has one copy. This splitting up of the two alleles is called segregation. Mendel bred yellow-seeded plants to green-seeded plants, and all of the offspring of the first generation had yellow seeds. None of them had green seeds. And he wanted to know if that genetic information for green seeds was still there in the plant somewhere, or if it had disappeared. So to find out, he crossed the offspring of that generation, and he looked at the second generation of offspring and saw that some of them did have green seeds. So this means that that genetic information of the recessive trait, it was still there and still being passed on. It was just hidden when that dominant yellow allele was present. So how did this happen? How did the recessive trait for green seeds disappear for an entire generation and then reappear in the next generation? Well, remember that each individual receives two copies of the gene, one from each parent. He took parents that only had green alleles and crossed them with parents that only had yellow alleles. And that way he knew exactly what the offspring would have. Every offspring from that cross got a green allele from one parent and a yellow allele from the other parent. So each one had both alleles, had one yellow and one green. And because yellow is dominant over green, every single one of those individuals expressed the yellow seed trait, not the green. But they still had that green allele in them. In the next step, he took individual plants that had both a yellow allele and a green allele. So he took plants from this generation that's outlined in red, and he bred them to each other, to other plants that had the exact same genetic makeup of a yellow allele and a green allele. And remember that segregation means that those alleles get split up when the plant reproduces. A parent plant doesn't give both alleles to the offspring, it only gives one to each offspring. So the dad plant give half of their offspring a yellow allele, but half of them a green allele. And the mom plants also gave half of their offspring a yellow and half of them a green allele. And because it's random, some of the baby plants got a yellow allele from each parent, some got a yellow from one parent and a green from one parent, and some got a green from each parent. And you can see the offspring that got a green from each parent are the only ones who actually had a green trait because it wasn't covered up by yellow. Okay, so here we have green and green, recessive and recessive. There's no capital A. There's no yellow to cover it up, so it was able to be green. Here, where we have a green, but then there's a yellow there, that yellow one is dominant, and so you can't even tell the green is there. There are terms to describe the setup of alleles that any individual has, and you need to know these terms. If they have two copies of a dominant allele, in this example, two alleles for yellow seeds, they are called homozygous dominant. If they have two copies of the recessive version of the allele, So for example, two alleles for green seeds, then they are homozygous recessive. And if they have two different alleles, in this example, one yellow and one green, they are heterozygous. Remember, the prefixes homo means the same and hetero means different. These terms describe the genotype of the individual. A genotype is the genetic makeup of the alleles. It doesn't describe what the individual looks like, it describes the alleles that it has. But we could use the genotype to figure out the phenotype. That's the trait that is expressed, okay, such as color. So you should notice that the heterozygous and homozygous dominant individuals have different genotypes, but the same phenotypes. Okay, so for our examples up here, our heterozygous dominant has two yellow alleles, and our heterozygous has one yellow and one green. But they have the same phenotype. They're both yellow, and it's because they both have that dominant yellow one in there. So this one has no choice but to be yellow as two yellow alleles, but this one, the heterozygous, has one yellow, which is dominant over the green. So all you see is yellow. And then homozygous recessive has a different phenotype. It has the phenotype that's green because it only has green alleles. It has nothing else. So there's more to go over, but we're going to just pause for a minute and do a review here. So again, if you're here live, I'm going to pop a question up on the screen for you to answer. For each gene, you have a copy from your mom and from your dad in every cell of your body, except each reproductive cell only ends up with one copy. So the separation of these two alleles in the reproductive cells is called what? We got 4 words to choose from there, so just there we go. So you should now see the poll on the screen. So if you're here live, you should answer it. I'd like to see all of our attendees answer the question before I move on, so I'll give you a few seconds to think about it. Okay, I got votes from half of you. I'm still missing a few people. Awesome, thank you. Okay, so I got answers from everybody. Okay, so I think you can see the answers, and again, I have no idea who answered what. Half of you said independent assortment, half of you said phenotypes, and or 25% said phenotypes and 25 segregation. So that kind of tells me we need to go back to that term. Make sure we're sharing the screen still. Okay, so the term for that is segregation. So remember, in all of your cells of your body, except for eggs and sperm, there are 2 of every gene. So for every gene, you inherited 1 copy from your mom and 1 copy from your dad. But it wouldn't make sense if you reproduce to give your offspring 2 copies, because the other parent is going to give 2 copies, and then it's going to have 4 copies, and then when they reproduce, it have 8. So that would be kind of crazy after a while. So what happens is they split up in those reproductive cells. And that splitting up is called segregation. So when we're talking about 2 copies of the same gene splitting up, the answer is segregation. So we go back to our question here. Okay, so the first 2 are just kind of unrelated words that are also from this section. Segregation is the correct answer. And a lot of people said independent assortment, and that's something we're going to learn about in a minute. Um, I'm wondering if these are people that already read this section and they thought it was similar. Independent assortment is going to be something we're talking about multiple genes. So you have genes that are kind of unrelated, maybe one for, well, examples we use is one for height and one for color. They're completely unrelated things. So those are going to be dealing with independent assortment. When we're talking about two copies of the same gene, so in this case, two copies of genes for color, maybe the yellow and the green and the gray genes for colors are different alleles, that's going to be segregation. Um, this is another review question, but I can't put it up as a poll because of the type of question it is, so just think about your answer. Match the genotype on the left to the description of what that type is on the right. So instead of using the letter A, we're using the letter X, but remember capital is still dominant and lowercase is recessive. So we have capital X, lowercase x, then we have two lowercase x's, and we have two capital X's. So think about what those are. One of them is homozygous dominant, one of them is heterozygous, one of them is homozygous recessive. So I'll give you about 10 seconds to think about it before I show you the answers. So a capital X and a lowercase x. So we have a capital and a lowercase, one of each. That's going to be heterozygous. Remember, hetero means different. So you have two different alleles. The other two are homozygous. Homo means same. So you have homozygous recessive with the lowercase and homozygous dominant with the uppercase. And remember, all of these are describing genotypes. Genotype is this. It's just the setup of the alleles. Okay, not phenotypes. Phenotype means actually what trait you see. Something like what color it is or how tall it is. All right. Now, jumping into the next thing. Due to this principle of dominance, where you have dominant and recessive alleles, we can figure out the probability of any trait showing up in offspring if we know the genotype of the parents. Now, the way that we demonstrate this is with a Punnett square. Okay, so that's the name for this diagram. You see them in your textbook. We figure out the parent's genotype, segregate the alleles, and put the alleles for one parent across the top and one down the side. So our genotype for one parent is capital A, little a. Now remember I said at the beginning capital A is going to be yellow, lowercase a is going to be green. So we split them apart, we segregate them, and put each one across the top. So basically these are our options. If you're inheriting a gene, you could inherit a capital A or you could inherit a lowercase a. Then we do the same thing down the side. We have capital and lowercase. It just happens to be the same. And that's what we have as options down the side for this plant. You could inherit capital or lowercase. Now that we have our edges filled out with the parents, so when you have a Punnett square, the top row and the side row are the parents, and then the rest of the chart is going to be the offspring. So now that we have all of that across the top and the side, we can find all the possible combinations of the offspring. Simply write the allele from the top of the column in all the squares in that column, and then write the allele at the left of each row in every square in that row. And now you're done. And this is every possible combination of alleles that could happen in the offspring of these two plants. you will see that some combinations appear more than once. So for example, we have a capital A and a little a, a capital A and a little a. So if they appear there more than once, then they have a higher percentage of the offspring that will have that genotype. So let's look at some numbers. Um, in this example, and this is the same example that we just saw, I just moved it to the next slide. A heterozygous plant crossed with a second heterozygous plant. 1 out of 4 of the offspring will get a homozygous dominant genotype. Okay, so sorry, there we go. 1 out of 4 will have that genotype. Squares here, four squares of offspring, only one of them was homozygous dominant. 2 out of 4, or 50%, will be heterozygous, and 1 out of 4 will be homozygous recessive. Now remember, that's the genotypes, but the phenotypes are different. Phenotypes is the color that we actually see. So 3 out of 4 have at least one dominant allele, and so 75% of the offspring, 3 out of 4, will be yellow because they have that dominant allele. Only 25% will be green, because only 25% have no dominant allele, just the recessive allele. So as a general rule, you're almost always going to end up having many more individuals with the dominant trait than the recessive trait. And you got to remember that the genotypes are not the same as the phenotypes. So our genotypes for this example, and they're different for every example, but for this example, our genotypes were 25%, 50%, and 25%. But our phenotypes are different. We've only got 2 phenotypes. We've either got yellow or green. There's nothing in between. So you got 75% and then 25% are green. And one more question that was answered by Mendel's work was whether traits stick together. If he had tall plants with green seeds and he had short plants with yellow seeds, what would happen if he crossed them? Would the height and color traits stick together? Would it be possible to get tall offspring with yellow seeds? The answer is that most traits are able to be split up and shuffled. And this principle is called independent assortment. The genes for height and color can be split up. So you can end up having a tall plant with yellow seeds because you don't have to inherit both traits from the same parent together. Okay, and this is something you could see in people. This is why you could inherit, your father's nose but your mother's eyes, because they're independently assorted. You don't necessarily get all the same genes as other offspring do. So your sister and brother might have a different set of features inherited from the parents because they got different combinations of genes. So we're going to show independent assortment for genes with height and color. And these are the alleles that we're going to use. Height will have a capital T for tall, which is the dominant trait, and a lowercase t for short, which is the recessive trait. Color will have a capital A for yellow, which is the dominant trait, and a lowercase a for green, which is the recessive trait. And we will start an example on the next slide that has both parents that are heterozygous for both traits. So if this is the mom plant here, okay, we've got a mom and a dad. So if the mom plant will have both a tall allele and a short allele, so it's heterozygous, it's got both. And then it's also going to have both a yellow allele and a green allele. So it's heterozygous for height here, and it's heterozygous for color. And then the dad plant will have the exact same setup. So this is just the example that we're going to do. And independent assortment of this can be shown within bigger Punnett square. And what you need to do to start is have every possible combination of alleles from the dad plant on the top and the mom plant down the side. So across the top, those combinations are tall and yellow, short and yellow, tall and green, and short and green. Then you put every possible combination of alleles from the mom down the side, and in this case they will be the exact same options. Tall and yellow, short and yellow, tall and green, short and green. Now, just like before, we fill them in down the columns. Okay, so everything from the top column, you just fill in into every block under it. And same thing across the rows. Everything in the first row gets that, and then it's just the same for every row. So filling them in down the columns and across the rows like this gives every possible combination of alleles of the offspring. Now, the phenotypes for this is more complicated when you're looking at two traits because now there's different combinations that we could have of height and color. So having both dominant traits, tall and yellow, is the most common, with 9 of these example offspring having at least one tall gene and one yellow gene. So remember, these are the dominant traits, so they only need one. You only need one capital letter for color to get yellow, and you only need one capital letter for height to get tall. So there's a total of 9 of these that have that. The next possibility is to have one dominant trait and one recessive trait. So 3 of the example offspring will be tall but have green seeds. So tall is, oops. Okay, well, 3, I don't know what happened with my examples there. Okay. Okay. 3 of the examples will be tall and have green seeds, but 3 of them will be short but have yellow seeds. So both of these are examples where you have one dominant trait. In this case, the dominant is yellow and the recessive trait is short. Or in this case over here, the dominant trait is tall and the recessive is green. So both of those have 3 of each. So you've got both cases where you have one dominant trait and one recessive trait. And out of all of our options, you get 3 that are short and yellow and 3 that are tall and green. And then the most rare possibility is having 2 recessive traits. So if there's only one example of offspring that is both short and has green seeds. Okay, so this is the only block where there's no dominant traits to block out those recessive traits. So whenever 2 parents that are heterozygous for both traits, so that's what we've got going on up here, heterozygous for both traits, heterozygous for both traits. Whenever that's the case and you cross those 2 parents, you're always going to end up with this ratio for the alleles of 9 to 3 to 3 to 1. So you need to remember that 9, where they are both dominant, is the most common. It's very rare to have one where they're both recessive, so that's why it's only 1. And then in between are the 2 versions that have one dominant trait and one recessive trait, and those are 3. So out of these 16 offspring examples, you get 9, 3, 3, and 1. Okay, so review questions again. Different genes are not usually inherited together. They get shuffled into different combinations. This is called what? Let me just bring up that poll. All right, so different genes, so completely separate genes, things like color and height, not related at all. Different genes are not usually inherited together. They get shuffled around. So what is the term to describe the genes getting shuffled around like that? It looks like someone just joined recently. That's okay. You can answer this question. I can't see your individual answers, just the group's answers. And I need more people to vote. So everybody think about it, choose your answer. I'm going to give you another 10 seconds or so. Okay, so not everybody voted, but at least I got some of you too. Um, but not two-thirds of you got the wrong answer and one-third of you got the right answer. So the right answer here is independent assortment. Different genes get shuffled around when they're inherited. They are not inherited together. Okay, again, you can inherit your mom's eyes and your dad's nose and your mom's ears and your dad's lips. Like it's just everything getting shuffled around. In plants, we're talking about height and color. Um, those two things get split up and shuffled around in the offspring so that we get something like this, where we have 16 different ways that they can get shuffled up, and we end up having 4 different phenotypes that you can get. Okay, so that happens due to independent assortment. Okay, those genes are independent of each other. So a lot of people said principle of dominance. That's not what this is. So the principle of dominance is that one gene will be dominant over another, but that has nothing to do with how they're sorted and split up in the offspring. And also, nobody said segregation. That's good. Segregation is when we're only talking about 2 alleles from the same gene. So you have 2 alleles for color. Um, they get split up when the organism reproduces. And one more review question. So when we're talking about making Punnett squares like we just did, if we have a parent, so let's say we have a mom plant and a dad plant, and this is our mom plant, and its genotype is this, capital A, capital B, capital B. When we go across the Punnett square, we're going to find every combination we can of those letters. So which one of these would not show up in that Punnett square? So I'll give you a chance to think about that and answer that question. Okay, I'd like to see some more votes. Ooh, almost everybody voted. I'll give you another 5 seconds for the last people. Okay, so the vote on this one is pretty split, so we're going to go over this a little bit. Um, I know this is hard to do without writing it all down, and you've maybe never seen these Punnett squares before. So it's going to look like this. So when we have 2 traits like this, we have an A and a B trait. the row on the top, okay, for one parent, has to have every combination of A's and B's that you can have. So if you take the capital A and the capital B, that goes here. You take the another capital B, because there is no B. You could have a lowercase a and a capital B, and then a lowercase a and another capital. Three of our options are exactly the same, but that's okay. That means that's going to affect what percentage of it. You can kind of think of it when I say that we need to have every combination. So we have A, B. Okay, so when I say we need to have every combination, it's okay that we're repeating some of these because we're looking at combinations like saying just the first one and the second one. So we have the first two, that's one combination. The second two, that's one combination. The outside ones, so the first A and the last B, and then the inside ones. So that's four different combinations we could get. So for our actual question, this is capital A, capital B is definitely one that we'll actually have. Little a, capital B is one that we'll have. And our third option we can't have. So that's the one that's not going to be in the Punnett square because there is no little b. So we can't have a little b because it doesn't exist. We only have capital B's. So this is different than the examples that we just went over, but it's just another example of a type of parent that you could have that goes into these Punnett squares. Okay, so I just want to review some key points from these sections. Traits are inherited because genes are transferred from parents to offspring. And different versions of a gene are called alleles. An individual has two copies of every gene, so they have two alleles, and they could be two different alleles or two of the same allele. Having two different alleles is called being heterozygous, and two of the same are called homozygous, and it's either homozygous dominant or homozygous recessive. Traits from recessive alleles are only seen if there is not a dominant allele present. They appear in homozygous recessive individuals only. They don't appear in heterozygous individuals because the dominant allele covers them in the heterozygous individuals. Two copies of a gene segregate when egg and sperm cells are produced so that each parent only gives one copy of the gene to offspring. An independent assortment means that most unrelated genes are not passed down together. A parent gives each offspring different random combinations of the genes. And that brings us to the end of this section. Come back next week for the next half of Chapter 11. And if you're not here live but you want some help, here are some ways that you can contact your instructors.